There are many theories in science: the Big Bang Theory, cell theory, and the general relativity theory. And scientific theories are different from any regular theory. But what is a scientific theory? A scientific theory is not an educated guess, but an explanation of a scientific event supported by scientific evidence. It has to be testable and tested over and over to confirm it as true, much like the endosymbiotic theory. What is the endosymbiotic theory? This theory gives an explanation for how eukaryotic cells could have evolved from prokaryotic cells that lived in symbiosis. Now, what prokaryotic symbiosis means is that these organisms live together.
Differences between endosymbiotic theory 1 and 2. Endosymbiotic theory 1 explains the origin of mitochondria, a primitive eukaryotic cell engulfed in aerobic prokaryote, likely an alpha protectobacterium, which evolved into the mitochondrion, responsible for energy production. Endosymbiotic theory 2 describes the origin of chloroplast, a eukaryotic cell with the mitochondria and engulfed a photosynthetic prokaryote, likely a cyanobacterium, which evolved into the chloroplast responsible for photosynthesis. Scientists believe this process happened twice because mitochondria and chloroplasts have 1. Distinct DNA resembling their prokaryotic ancestors, 2. Double membranes consistent with engulfment, and number 3. Prokaryotic-like ribosomes different from eukaryotic ribosomes.
The endosymbiotic theory proposes that organelles like mitochondria and chloroplasts originated from free-living bacteria that were engulfed by another cell, eventually forming a symbiotic relationship. This theory revolutionized our understanding of evolution and the origin of complex life. The societal implications are vast. For instance, the discovery that mitochondria have their own DNA has led to research on mitochondrial diseases and potential therapies, including gene editing techniques. Additionally, the idea of symbiosis inspires innovative approaches in biotechnology, such as using engineered bacteria for medical treatments or environmental solutions. A historical metaphor for this theory could be the merging of cultures or empires, like the Roman Empire incorporating various people and their practices to form a stronger, unified entity. Similarly, their partnership between the host cell and bacteria led to more complex and efficient life forms.
These are the benefits to both an anaerobic cell and an aerobic cell. Starting with the benefits to an anaerobic cell. First, the anaerobic host cell provides physical protection in a relatively stable internal environment, shielding the aerobic cell from environmental fluctuations and predators. Second, the host cell supplies nutrients and organic molecules that the aerobic cell oxidizes for energy production. Third, living inside the host cell reduces external stresses such as changes in temperature or pH, allowing the aerobic cell to maintain optimal functionality. These are the benefits to the aerobic cell. First, the aerobic cell performs oxidative phosphorylation, producing significantly more ATP than aerobic glycolysis. This surplus energy enhances the host cell's survival and functionality. Second, by metabolizing oxygen, the aerobic cell detoxifies the environment, reducing oxidative damage and enabling the host cell to survive in oxygen-rich conditions. Third, the increased energy production and protection from oxidative stress confer a competitive advantage, enhancing the host cell's reproduction and survival in this environment. Overall, this mutualistic relationship ultimately led to the evolution of mitochondria within eukaryotic cells, marking a pivotal step in the development of complex life forms.
These are the 5 similarities between mitochondria and chloroplasts and bacterial cells. Energy transfer. Both mitochondria and chloroplasts act as centers for energy transformation in cells, similar to how bacterial cells utilize metabolic processes. The double membrane structure. Both organelles have double membranes, a feature shared with bacterial cells due to their likely prokaryotic origin. Own DNA. Mitochondria and chloroplasts possess their own circular DNA, which resembles bacterial DNA. Self-replication. Both organelles can replicate independently of the cell, akin to bacteria's binary fission. Endosymbiotic origin. Both are believed to have evolved from free-living prokaryotic cells, supporting the endosymbiotic theory.
While there's two lipid bilayers around both the mitochondria and chloroplast,
The mitochondria and chloroplasts have double membranes because they originated from free-living prokaryotes according to the endosymbiotic theory. The outer membrane is derived from the host cell's engulfing membrane, while the inner membrane comes from the original prokaryotic. The double membrane system enhances metabolic functions in several ways. Number 1. Increases surface area. The inner membrane has a large surface area for embedding proteins involved in processes like the electron transport chain and ATP synthesis. Number 2 is compartmentalization, which is the double membrane creates distinct compartments such as inner membrane space in the matrix in the mitochondria and inner membrane space and stroma in the chloroplast, allowing different reactions to occur simultaneously in optimized environments. Number 3 is selective transport. The membranes control transport of molecules in and out of the organelle through protein channels and pumps, ensuring the regulation of the internal environment. Number 4 is proton gradient formation. The inner membrane is essential for establishing and maintaining proton gradients during the ATC, which drives ATP synthesis through hemiosmosis. This structure allows mitochondria and chloroplasts to perform energy metabolism efficiently and adaptively within the eukaryotic cell.